Today we discuss functions of protein. As you all know, collection of proteins within a cell determines its health and functions. Proteins are responsible for every task of cellular life, including cell shape and inner organization, product manufacture, and waste cleanup and routine maintenance. Proteins also receive signals from outside the cells and mobilize intracellular response. They are the workhorse macromolecules of the cell and are as diverse as the functions they serve. Protein is crucial to good health. In fact, the name comes from the Greek word proteos, meaning primary or first place. Proteins are made up of amino acids that join together to form long chains. You can think of a protein as a string of beads in which each bead is an amino acid. There are 20 amino acids that help form the thousands of different proteins in your body. Proteins do most of their work in the cell and perform various jobs. Proteins can be big or small, mostly hydrophobic or hydrophilic, exist alone or as part of a multi-unit structure, and change shape frequently or remain virtually immobile. All of these differences arise from the unique amino acid sequences that make up proteins. Fully folded proteins also have distinct surface characteristics that determine which other molecules they interact with. When proteins bind with other molecules, their conformation can change in subtle or dramatic ways. Protein functions are as diverse as protein structure. For example, structural proteins maintain cell shape akin to a skeleton, and they compose structural elements in connective tissues like cartilage and bone in vertebrates. Enzymes are another type of protein, and these molecules catalyze the biochemical reactions that occur in cells. Yet other proteins work as monitors, changing their shapes and activity in response to metabolic signals or messages from outside the cells. Cells also secrete various proteins that become part of the extracellular matrix or are involved in intracellular communication. Proteins are sometimes altered after translation, and folding are complete. In such cases, transferase enzymes add small modifier groups such as phosphatase or carboxylic groups to the proteins. These modifications shift the protein conformation and act as molecular switches that turn the activity of a protein on or off. Many post-translational modifications are reversible, although different enzymes catalyze the reverse reaction. For example, enzymes called kinases add phosphate groups to the proteins, but enzymes called phosphatases are required to remove these phosphate groups. Whenever the body is growing, repairing, or replacing the tissues, proteins are always involved in these reactions. Sometimes their roles to facilitate or regulate, other times it is to become part of a structure. Versatility is a key feature of proteins. From the conception, Proteins form the building blocks of muscles, blood, and skin. In fact, most body structures. Proteins form the most body structures. For example, to build a bone or a teeth, cell first lay down a matrix of the protein collagen and then fill it with crystals of calcium, phosphorus, magnesium, fluoride, and other minerals. Collagen also provides the material of ligaments and tendons and the strengthening glue between the cells of the artery walls that enables the arteries to withstand the pressure of the blood surging through them with each heartbeat. Also made of collagen are scars that knit the separated parts of torn tissues together. Proteins are also needed for replacement. The lifespan of a skin cell is only about 30 days. As old skin cells are shed, new cells made largely of protein grow from underneath to compensate. Cells in the deeper skin layers synthesizes new proteins to go into hair and fingernails. Muscle cells make new proteins to grow larger and stronger in response to exercise. Cells of the GIT tract are replaced every 3 days, both inside and outside. Then the body continuously deposits protein into new cells that replace those that have been lost. Body needs protein for growth and maintenance of tissues. Your body's proteins are in a constant state of turnover. Under normal circumstances, your body breaks down the same amount of protein that it uses to build and repair tissues. Other time, it breaks down more protein than it can create, thus increasing your body's need. This typically happens in periods of illness during pregnancy and while breastfeeding. People recovering from an injury or surgery, older adults, and athletes require more protein as well. Collagen contains specific amino acids: glycine, proline, hydroxyproline, and arginine. These amino acids have a regular arrangement in each of the three chains of these collagen subunits. Proline or hydroxyproline constitute about one-sixth of the total sequence. Glycine is found at most every third residue. Glycine accounts for one-third of the sequence, meaning that approximately half of the collagen sequence is not glycine, proline, or hydroxyproline. Proline makes up about 17% of collagen. Collagen also has two uncommon derivative amino acids that are not directly inserted during translation. These amino acids are found at specific locations relative to glycine and are modified post-translationally by different enzymes, both of which require vitamin C as a cofactor. Hydroxyproline is derived from proline and hydroxylysine derived from lysine. Depending on the type of collagen, varying numbers of hydroxylysines are glycosylated, mostly having disaccharides attached. The regular repetition and high glycine content is found in only a few other fibrous proteins, such as silk fibrin. In silk, 75 to 80% is glycine, alanine, glycine, alanine, with 10% serine, and elastin is rich in glycine, proline, and alanine, whose side group is a small inert methyl group. High glycine contents are not found in globular proteins, except in very short sections of their sequence. Because glycine is the smallest amino acid with no side chain, it plays a unique role in fibrous structural proteins. Cortisol stimulates the degradation of skin collagen into amino acid. During the translation, two types of the peptide chains are formed on ribosomes along the rough endoplasmic reticulum. These are called alpha 1 and alpha 2 chains. These peptide chains, known as preprocollagen, have registration peptides on each and a single peptide. The preprocollagen is released into the lumen of the RER. The signal peptide are cleaved inside and the peptide chains are new called pro-alpha chains. Hydroxylation of lysine and proline amino acids occur inside the lumen. This process is dependent on ascorbic acid as a cofactor. Further glycosylation of specific hydroxylysine residues occur. Triple helical structure is formed inside the endoplasmic reticulum from each two alpha chains and one alpha 2 chains. This is called procollagen. Procollagen is transported into the Golgi of Bristles, where it is packed and secreted by the exocytosis. Once outside the cell, the registration peptides are cleaved and tropocollagen is formed by the procollagen peptidase. These tropocollagen molecules gather to form collagen fibrils via covalent cross-linkages by lysyl oxidase, which links the hydroxylysine and lysine residues. Multiple collagen fibrils form into collagen fibrils. Collagen may be attached to cell membranes via several types of proteins, including fibrinolactin and integrin. Vitamin C forms an important component of the process of collagen synthesis. Vitamin C deficiency causes scurvy, a serious and painful disease in which the collagen that is synthesized is defective and it does not produce strong connective tissues. This leads to bleeding and peeling gums, loss of teeth, skin discoloration, and non-healing wounds. Prior to the 18th century, this condition was notorious among long-duration naval and military expeditions, during which participants were deprived of foods containing vitamin C. Certain autoimmune diseases such as systemic lupus erythematosus or rheumatoid arthritis may occur where the body's immune system perceives the collagen as foreign and attacks and degrades the collagen in the body. Some bacteria and viruses also destroy the collagen fibers in the body or interfere with its production. Some proteins function as enzymes. Proteins that catalyze the specific biochemical reactions. Enzymes facilitate biochemical reactions and speed them up, making them as much as a million times faster. There are thousands of enzymes, and each type facilitates a specific biochemical reaction. Enzyme only acts on specific reactant molecule that is substrate to produce a specific end product or products. These rely on thousands of different enzymes to catalyze metabolic reactions. Enzymes are basically proteins and they make a biochemical reaction more likely to proceed by lowering the activation energy of the reaction, making these reactions proceed thousands or even millions of times faster than they would without a catalyst. Enzymes are highly specific to their substrates. They bind these substrates at complementary areas on their surfaces, providing a snug fit that may compare to a lock and key. Enzymes work by binding one or more substrates, bringing them together so that a reaction can take place and releasing them once the reaction is complete. When substrate binding occur, enzymes undergo a conformational shift that orients or strains the substrates so that they are more reactive. The name of an enzyme usually refers to the type of biochemical reactions it catalyzes. For example, proteases break down protein, and dehydrogenases oxidize a substrate by removing hydrogen atoms. As a given rule and the journal rule, the "ase" suffix identifies a protein as an enzyme, whereas the first part of an enzyme name refers to the reaction that it catalyzes. The proteins in the plasma membrane typically help the cell interact with its environment. For example, plasma membrane protein carry out functions as diverse as ferrying nutrients across the plasma membrane, receiving chemical signals from outside the cell, translating chemical signals into intracellular actions, and sometimes anchoring the cells in a particular location. The overall surfaces of the membrane proteins are mosaics, with patches of hydrophobic amino acids where the proteins contact lipids in the membrane bilayer, and patches of hydrophilic amino acid on the surfaces that extend into the water-based cytoplasm. Many proteins can move within the plasma membrane through a process called membrane diffusion. This concept of membrane-bound proteins that can travel within the membrane is called the fluid mosaic model of the cell membrane. The portions of the membrane proteins that extend beyond the lipid bilayer into the extracellular environment are also hydrophilic and are frequently modified by the addition of sugar molecules. Other proteins are associated with the membrane but not inserted into it. They are sometimes anchored to lipid in the membrane or bound to the other membrane proteins. Switch their protein machinery on or off in response to the body's need. Hormones do the switching with marvelous precision. The body's many hormones are messenger molecules, and some hormones are proteins. Some hormones that are products of endocrine glands are proteins or peptides. Others are steroids. The origin of hormone, their physiological role, and their mode of action are dealt with the hormone. None of the hormones has any enzymatic activity. Each has a target organ in which it elicits some biological actions, that is, secretion of gastric or pancreatic juice, production of milk, production of steroid hormones. The mechanism by which the hormone exerts their effect since 1949. Repeated attempts to synthesize it gave very poor yields because of the failure of the two peptide chains to combine, forming the correct disulfide bridge. The ease of the biosynthesis of insulin is explained by the discovery in the pancreas of proinsulin, from which insulin is formed. The single peptide chain of proinsulin loses a peptide consisting of 33 amino acids and called the connecting peptide or C-peptide. During its conversion to insulin, the disulfide bridges of proinsulin connects the A and B chains. The pituitary gland has an anterior lobe or a posterior lobe and an intermediate portion. They differ in cellular structure and in the structure and action of the hormones they form. The posterior lobe produces two similar hormones, oxytocin and vasopressin. The former causes the contraction of the pregnant uterus, the latter raises the blood pressure. Both are formed by a ring of 5 amino acids. The two cysteine halves count as one amino acid, and a side chain of 3 amino acids. The two cysteine halves are linked to each other by a disulfide bond, and the C-terminal amino acid is glycinamide. Human vasopressins differ from oxytocin in that isoleucine is replaced by phenylalanine and leucine by arginine. The intermediate part of the pituitary gland produces the melanocyte-stimulating hormone, which causes the expansion of the pigmented melanophores in the skin of the Bechdel chains. Two hormones called alpha MSH and beta MSH have been prepared from hog pituitary glands. The anterior pituitary lobe produces these several protein hormones: a thyroid-stimulating hormone which is thyrotrophin, a lactogenic hormone, a growth hormone, and a luteinizing hormone, follicle-stimulating hormone. The thyroid-stimulating hormone consists of alpha and beta subunit with a composition similar to the subunits of luteinizing hormone. When separated, neither of the two subunits has hormonal activity. When combined, they regain about 50% of the original activity. The lactogenic hormone, prolactin, contain the 190 amino acid. Their sequences had been elucidated. A similar peptide chain of 188 amino acid that has been synthesized not only has 10% of the biological activity of the natural hormones. Hormone insulin provides a familiar example. When blood glucose rises, the pancreas releases its insulin. Insulin stimulates the transport proteins of the muscle and adipose tissues to pump glucose into the cell faster than it can leak out. As blood glucose falls, the pancreas slows its release of hormone, insulin. Many other proteins act as hormone regulating a variety of actions in the body. Glucagon, which signals the breakdown of stored glucose in the liver. Human growth hormone stimulate the growth of various tissues including bone. Antidiuretic hormone signals the kidneys to reabsorb water. Adrenocorticotropic hormone stimulates the release of cortisol, a key factor in the metabolism. Peptides. These are made from chains of amino acids, ranging from a few to several hundred. Steroids. These are made from the fat cholesterol. Testosterone and estrogen are steroid bases. Amines, these are made from the individual amino acids tryptophan or tyrosine, which help make hormones related to sleep and metabolism. The proteins in the plasma membrane help the cell interact with its environment. Plasma membrane proteins carry out functions as diverse as ferrying nutrient across the plasma membrane, receiving the chemical signals from outside the cell, translating chemical signals into the intracellular actions, and sometimes anchoring the cells in a particular location. The overall surface of the membrane proteins are mosaics, with patches of hydrophobic amino acids where the proteins contact lipids in the membrane bilayer, and patches of hydrophilic amino acids on the surface that extend into the water-based cytoplasm. Proteins serve a variety of functions within the cells. Some are involved in structural support and movement, others in enzymatic activity. The cytoplasm is highly structured due to the proteins, particularly in the eukaryotic cells, which tend to be bigger and need more mechanical support than prokaryotic cells. An extensive network of filaments, microtubules, actin filaments, and intermediate filaments can be detected with a variety of microscopic methods. Microtubules play a major role in organizing the cytoplasm and in the distribution of organelles. They also form the mitotic spindle during the cell division. Actin filaments are involved in various forms of cell movement, include cell locomotion, contracting of muscle cells, and cell division. Regulate body processes to maintain the fluid balance. Albumin and globulin are proteins in your blood that help maintain your body's fluid balance by attracting and retaining water. If you don't eat enough protein, your levels of albumin and globulin decreases. These proteins can no longer keep blood in your blood vessels and the fluid is forced into the spaces between your cells. As the fluid continues to build up in the spaces between your cells, swelling or edema occur, particularly in the stomach region. This is the form of the severe protein malnutrition called kwashiorkor that develops when a person is consuming enough calories but not consume enough protein. Protein helps to maintain the body's fluid balance. Extracellular fluid can be found either in the spaces between the cells, which is interstitial spaces, or within the blood vessels, which is intravascular spaces. The fluid within the intravascular spaces is called plasma. Fluid can flow freely between these compartments, but being large, proteins cannot. Proteins are trapped within the cells and to a lesser extent in the plasma. Wherever proteins are, they attract water. The exchange of materials between the blood and the cells take place across the capillaries wall, which allow the passage of fluid and a variety of materials, but usually not plasma proteins. Some plasma proteins leak out of the capillaries into the interstitial fluid between the cells. These proteins cannot be reabsorbed back into the plasma. They normally re-enter circulation via the lymph system. If plasma proteins enter the interstitial spaces faster than they can be cleared, fluid accumulates because proteins attract the water and causes the swelling. Swelling due to an excess of interstitial fluid is known as edema. Excessive protein losses caused by kidney diseases or large wounds, such as extensive burns. Inadequate protein synthesis caused by liver disease. Inadequate dietary intake of proteins. Whatever the cause of edema, the result is the same: a diminished capacity to deliver nutrients and oxygen to the cells and to remove waste from them. As a consequence, cells fail to function adequately. Proteins plays a vital role in regulating the concentrations of acid and bases in your blood and other bodily fluids. The balance between acids and bases is measured using the pH scale. It ranges from 0 to 14, with 0 being the most acidic, 7 neutral, and 14 the most alkaline. Some examples of the pH value of common substances include the stomach pH is 2, tomato juices have the pH value of 4, black coffee has the pH value of 5, human blood has the pH value 7.4, and soapy water contain the pH 12. A variety of buffering systems allows your bodily fluids to maintain normal pH ranges. A constant pH is necessary, as even a slight change in pH can be harmful or potentially deadly. One way your body regulates the pH is with proteins. An example is hemoglobin, a protein that makes up red blood cells. Hemoglobin binds small amounts of the acids, helping to maintain normal pH value of your blood. The other buffer system in your body includes the phosphate and bicarbonate. Normal body processes continually produces the acids and bases, which the blood carries to the kidneys and lungs for excretion. The challenge is to do this without upsetting the blood's acid-base balance. In an acid solution, hydrogen ions abound. The more hydrogen ions, the more concentrated the acid. Proteins which have negative charges on their surfaces attract hydrogen ions which have positive charges. By accepting and releasing the hydrogen ions, proteins maintain the acid-base balance of the blood and the body fluids. The blood's acid-base balance is tightly controlled. The extremes of acidosis and alkalosis leads to coma and death, largely because they denature working proteins. Disturbing a protein's shape renders it useless. To give just one example is the denatured hemoglobin loses its capacity to carry oxygen. Blood pH is maintained between 7.35 and 7.45, which is slightly basic. Even a slight change in blood pH can affect your functions. The body has several systems that hold the blood pH within the normal range to prevent this from happening. One of these is circulating albumin. Albumin is slightly acidic, and because it is negatively charged, it balances the many positively charged molecules such as hydrogen protons, calcium, potassium, and magnesium, which are also circulating in the blood. Albumin acts as a buffer against abrupt changes in the concentrations of these molecules, balancing blood pH and maintaining the status quo. The protein hemoglobin also participates in acid-base balance by binding hydrogen protons. Transport proteins carry substances throughout your bloodstream into cell. out of cells or within the cells. The substances transported by these proteins included nutrients like vitamin or mineral, blood sugar, cholesterol, and oxygen. For example, hemoglobin is a protein that carries the oxygen from your lungs to body tissues. Glucose transporters move glucose to your cells, while lipoproteins transport cholesterol and other fats in your blood. Protein transporters are specific, meaning they will only bind to specific substances. A protein transport that moves glucose will not move cholesterol. Proteins also have storage roles. Ferritin is a storage protein that stores iron. Another storage protein is casein, which is the principal protein in milk that helps babies grow. Transport of the mineral iron provides an good illustration of these proteins' specificity and precision. When iron enters intestinal cells after a meal has been digested and absorbed, it is captured by a protein. Before leaving the intestinal cell, iron is attached to another protein that carries it through the bloodstream to the cell. Once iron enters a cell, it is attached to a storage protein that will hold the iron until it is needed. When it is needed, iron is incorporated into the proteins in the red blood cells and muscles that assist in oxygen transport and use. Some transport proteins reside in cell membrane and act as a pump, picking up compounds on one side of the membrane and releasing them on the other as needed. Each transport protein is specific for a certain compounds or group of related compounds. These help to maintain sodium and potassium concentrations in the fluid inside and outside the cell. The balance of these two minerals is critical to nerve transmission and muscle contraction. Imbalances can cause irregular heartbeat, muscular weakness, kidney failure, and even death. Proteins also defend the body against disease. A virus, whether it is one that causes the flu, smallpox, measles, or the common cold, enters the cell and multiplies there. One virus may produce 100 replicas of itself within an hour. Each replica can then burst out and invade 100 different cells, yielding 10,000 virus particles, which invade 10,000 cells. Left free to do their worst, they will soon overwhelm the bodies with disease. When the body detects these invading antigens, it manufactures the antibodies, giant protein molecules designed specifically to combat them. The antibodies work so swiftly and efficiently that in a normal, healthy individual, most diseases never have a chance to get started. Without sufficient protein, the body cannot maintain its army of antibodies to resist infectious diseases. Each antibody is designed to destroy just one antigen. Once the body has manufactured antibodies against a particular antigen, it remembers how to make them. The next time the body encounters that same antigen, it will produce antibodies even more quickly. The body develops a molecular memory known as immunity. Without these antibodies, bacteria and viruses would be free to multiply and overwhelm your body with the disease they cause. Once your body has produced the antibodies against a particular bacteria, your cell never forget how to make them. As a result, your body develop immunity against the diseases to which it is exposed. Antibodies which are produced in response to the antigen, which is the foreign invaders in your body, these antibodies are protein in nature in your blood that help protect your body from the harmful invaders. Protein helps form the immunoglobulin or antibodies to fight the infection. Can supply your body with energy. Protein contains 4 calories per kg, the same amount of energy that carbohydrate provide. Fats supply the most energy at 9 calories per gram. The last thing your body wants to use for energy is protein, since this valuable nutrient is widely used throughout your body. Carbohydrates and fat are much better suited for providing energy as your body maintains reserves for use as fuel. They are metabolized more efficiently compared to protein. Protein supplies your body with very little of its energy needs under normal circumstances. Instead of fasting 18 to 48 hours of no food intake, your body breaks down skeletal muscles so that the amino acids can supply you with energy. Your body also uses the amino acids from breakdown skeletal muscles if carbohydrate storage is low. This can occur after exhaustive exercise or if you don't consume enough calories in general. Protein provides the calories and energy in the time of the deprivation, if need be. Without energy, cells die. Without glucose, the brain and nervous system falter. Only about 10% of the dietary proteins are catabolized each day to make cellular energy. The liver is able to break down amino acids to the carbon skeleton, which can then be fed into the citric acid cycle. This is similar to the way that glucose is used to make ATP. If a person's diet does not contain enough carbohydrates and fats, their body will use more amino acids to make energy, which compromises the synthesis of new proteins and destroys the muscle protein. If a person's diet contains more protein than the body needs, the extra amino acid will be break down and transformed into the fat. Are involved in all aspects of wound healing, a process that takes place in 3 phases: inflammatory, proliferative, and remodeling. For example, if you were sewing and pricked your finger with a needle, your flesh wound turn red and become inflamed. Within a few seconds, bleeding would stop. The healing process begins with proteins such as bradykinin, which dilate the blood vessels at the site of injury. An additional protein called fibrin helps to secure platelets that form a clot to stop the bleeding. In the proliferative phase, cells move in and mend the injured tissues by installing newly made collagen fibers. The collagen fibers help pull the wound's edges together. In the remodeling phase, more collagen is deposited, forming a scar. Scar tissue is only about 80% as functional as normal uninjured tissues. If a diet is insufficient in protein, the process of wound healing is markedly slowed. Wound healing takes place only after an injury is sustained. A different process called tissue regeneration is ongoing in the body. The main differences between wound healing and tissue regeneration is in the process of regeneration, an exact structural and functional copy of the lost tissue. Old dying tissue is not replaced with the scar tissues, but with the brand new, fully functional tissues. Some cells such as skin, hair, nails and intestinal cells have a very high rate of regeneration, while others such as heart muscle cells and nerve cells do not regenerate at any appreciable levels. Tissue regeneration is the creation of new cell, that is cell division, which requires many different proteins including enzymes that synthesize the RNA and protein, transport proteins, hormones and collagen. In a hair follicle, cells divide and a hair grows in length. Hair growth averages 1 centimeter per month and fingernails about 1 centimeter every 100 days. The cell linings the intestine regenerate every 3 to 5 days. Protein inadequate diet impair tissue regeneration, causing many health problems including impairment of nutrient digestion and absorption, and more visibly hair and nail growth. Building block of your muscles. Eating adequate amounts of protein helps you maintain your muscle mass and promote the muscle growth when you do strength training. If you are physically active, lifting weights or trying to gain muscles, you need to make sure you are getting enough protein. Keeping protein high intake can also help prevent muscle loss during the weight loss. An ongoing myth, the idea that protein, mainly animal protein, is bad for your bones. This is based on the idea that protein increases the acid load in the body, leading to calcium leaching from your bones in order to neutralize the acid. People who eat more protein tend to maintain bone mass better as they age and have a much lower risk of osteoporosis and fractures. This is especially important for the women who are at risk of osteoporosis after menopause. Eating plenty of proteins and staying active is a good way to help prevent that from happening. A food craving is different from normal hunger. It is not just about your body needing energy or nutrients, but your brain needing a reward. Craving can be incredibly hard to control. The best way to overcome them may be to prevent them from occurring in the first place. One of the best prevention methods is to increase your protein intake. This may be mediated by an improvement in the function of the dopamine, one of the main brain hormones involved in craving and addiction. Eating can boost your metabolism for a short while because your body uses the calories to digest and make use of the nutrients in food. This is referred to as the thermic effect of food. Not all foods are the same in this regard. In fact, protein has a much higher thermic effect than fat or carbohydrates, which is 20 to 35% compared to 5 to 15%. Protein also participate in some of the body's most amazing activities such as vision. When a tissue is injured, a rapid chain of events lead to the production of the fibrin, a stringy insoluble mass of the protein fibers that forms a clot from liquid blood. The light-sensitive pigments in the cells of the retina are molecules of the protein opsin. Opsin respond to light by changing its shape, initiating the nerve impulses that convey the sense of the sight to the brain. Opsins are a G-protein coupled receptors, a group of proteins made light-sensitive via the chromophore retinal. They are found in photoreceptor cells of the retina. Five classical groups of the opsins are involved in vision, mediating the conversion of a photon of light into an electrochemical signal, the first step in the visual transduction cascade. Another opsin found in the mammalian retina, melanopsin, is involved in the circadian rhythms but not in visions. Humans have in total 9 opsins. Besides vision and light perceptions, opsins may also sense the temperature, sound, and chemicals.